Thank you very much. Is this an okay volume? Okay. So when they asked me to do this talk, I thought, oh, this is going to be easy. I'm going to just give one of my canned talks that we give all the time for hereditary cancer. And then I started getting emails from Jim and from Ethan. I'm reading some name tags, so I found you already, I think, with some really great questions. So I tried to revamp my slides and give you some information about what I focus on, which is hereditary cancer, in other words, cancer running in a family, but also talk a little about the hot topic of tumor genetic testing. And you may, if you're actively going through treatment or surveillance and working with an oncologist, this may be some terminology you've heard or you've read about. So I tried to include a little bit of that in my talk as well. And just like Jim said, please keep it informal. So if you think of a question, don't let it slip your mind. Go ahead and raise your hand and I'll try to catch you. So I have given a lot of information about myself to Jim. You covered my first two slides of who I am. So I'll skip those. But I will say, maybe if you could just raise your hand a little bit if you've heard of a genetic counselor before. So that's great. That's more than we would have heard of 20 years ago, right? So how I explain genetic counseling is we're kind of like a translator. So we take complex genetic information and try to explain it in a way with a lot of education, visual aids, or connections to online resources or other connection groups like this group here, where we connect people to better understand what's going on with their disease, with their cancer. Genetic counselors work in other fields too, childhood diseases, rare diseases, pregnant women. Just happens to be oncology is the area that I specialized in. So you'll have to bear with me. I have to go through my basic what is genes and chromosomes and genetics to make sure that I'm all on the same page as I explain some of these more complex parts. One thing I wanted to ask in getting to know the group a little bit more is if you could maybe raise your hand if you have a prostate cancer or know someone with prostate cancer. So I'm expecting 100%, right? Okay. So keep them up. Okay. Keep your hands up if you have a family member who has any type of cancer. Prostate, pancreatic, breast. Okay, so everyone's still pretty much raised. How about if you have a family member with prostate cancer as well? So think back to your brothers, your dad, your uncle. So you can lower them if you don't have anyone else with prostate cancer. There's a lot of people that are still have your hands raised, which means that, and you can lower them now, thank you. It means that the question has probably crossed your mind. Is my prostate cancer, is that the same cause as my brother, as my dad, as my uncle? And what could be causing that? And that's one of the main issues that we focus on every day in the patients that we work with. So a little about genetics as we're trying to answer this question. Genetics 101 is you have a cell, which is over there on the left side of your screen, right here. Inside is the nucleus, that's the center of the cell that is kind of like the brains of the cell. And inside of there, a structure is called chromosomes, and that holds all of a person's genetic material. You have 46 chromosomes in each cell. That's like thinking about a book, an encyclopedia, if you will. So you have 46 volumes of an encyclopedia, which are represented by these chromosomes. And if you take one chromosome and you stretch it out, kind of like a slinky, so you make it really long, that's where you get this strand of DNA. And then one section of DNA is what we call a gene. So this is one specific message in your body or in a cell so that your body can read that gene, almost like a sentence if you notice these letters, and it creates a protein product. So this whole process of your body knowing what gene to read and getting the correct message to create a protein is essential to life. I can give you an example. A very simple example would be you have a gene for eye color, let's say, or hair color. So your body reads this green eye color gene and creates a green protein that's then expressed in your eyes. You have people with blue eyes or brown eyes would have a slightly different genetic code, so that's what makes us all different. What can arise, though, is a problem with one of these genes. That could be a typo in your sentence. Misspelled letter, a missing piece of this gene, and that impacts the protein that has this downstream effect or function. So that's really crucial to understand for everything we talk about from here on out, because genetic mutations play a huge role in cancer. I wanted to show you a picture of chromosomes. So this is actual chromosomes that would be looked at under a microscope. They're not always this nicely organized. Usually they're a big gobbled mess, kind of like a bowl of spaghetti. But someone's job is to take these and separate them out, so individual chromosomes. And the reason we like to do this is to show that every cell has 2 copies of every chromosome. So if you'll notice, they're paired up and labeled 1, 2, 3, 4. So you have 23 pairs, and this also means that you have 2 copies of each chromosome, so 2 copies of number 3 or 2 copies of number 9. And what we know is that people inherit half of their chromosomes, or half of their genetic material from their mom and half from their dad. So hence, for each of these copies, you would have 1 copy from mom and 1 copy from dad. Remember that along these chromosomes, you have thousands and thousands of genes all lined up on that DNA strand, all having some type of function or role in your body. So when we talk about cancer, there's really 2 sets of genetics you have to keep in mind. You have your genetics that you were born with. So those are chromosomes that I just showed you, where you had a sperm and an egg. They came together, they each contributed half of your genetics. And that's what created the first cell that became a person, which then replicated to create us as adults. And we can check the DNA in your, that you originally were born with by doing a blood test. And so this, from the blood, they can take cells, white blood cells, identify the DNA, take those out and look at that to see what you inherited from your parents. But there's also another story to DNA that's going on, especially in cancer cells. So if we take a biopsy of a cancer, and in this case it's using a lung cancer as an example, but really any cancer would count for this. When we look at the genetics of a cancer cell, that cell or those tissues have a lot of genetic mutations that aren't present in your original DNA. And that's the premise of cancer. It's taking normal cells, and over time, or for many different reasons, those cells acquire genetic mutations to an endpoint where these cells don't even look like their original version. They have so many mutations, they're not doing their normal job as, let's say, a prostate cell, or a lung cell, or a breast cell. Instead, they've taken on the roles of a cancer cell. Are we doing good so far? Okay. So I'm going to skip this because you guys know this, statistics for prostate cancer, but what I wanted to show, talk a little bit about is the risk factors for prostate cancer, because genetics is just one of those parts. So the biggest risk factors for prostate cancer would be age, over age 50, and that's a clue that tells us that time is important. For your normal cells to transform into a cancer cell, it takes years, usually. Race is important, ethnicity. So what we know is that the gradient of risk for prostate cancer especially is highest for African American, kind of European Caucasian is middle of the road, and then the lowest risk is in Native American and Asian populations. So why is that? There's some genetic link there, and there could also be some shared environment or culture that goes into that, but mainly we think it's a genetic risk. And then finally, family history. So what we know is that when they first started studying genetics and they looked at families, what they found is that the risk for one man to develop prostate cancer was higher if his dad or his brother had prostate cancer first. So it's showing that there's some genetic link passed on within a family in some or maybe a majority of cases. So I like to show this table because often at this point someone raises their hand and says, what's my risk for prostate cancer if my 2 brothers have already had it, or something to that effect. And we can kind of use this table as a guide. It also shows that the more family history of prostate or other cancers there are in your family, the higher the risk is. So you could also think of this as a way if you have sons, what their risk might be. So example, no family history of any prostate cancer, about a 1 in 6 risk. brother or father with prostate cancer at any age, so even if they're 90 years old and develop prostate cancer, that's going to slightly, or sometimes not so slightly, increase their risk for prostate cancer in the other men in the family. And then it goes on to say if you have multiple family members, so a father and a brother, now we're looking at a much higher risk. And then finally, this last slide, or the last part of the table, is the hereditary prostate cancer. So hereditary prostate cancer, and we'll talk a lot more about this, is when there's a very specific known genetic mutation that is defective in a person's body, that's inherited from one generation to the next, that increases their risk for cancer. And so with prostate cancer, the risk can be as high as getting close to 50% if we know someone is a gene mutation carrier, as we call it. But there are other risk factors too, and really none of these risk factors work in isolation. So there's hormonal levels, environment, so your diet, BMI, smoking history, alcohol use, what supplements someone takes. And really the effect of all of these is maybe small individually, but by the time someone has multiple risk factors, that's where maybe we see an additive effect in the risk for prostate cancer. And this is my slide just to remind myself to tell you that it's not just genetic risk factors, but it's really a combination of risk factors. Bear with me here. I wanted to go through how we classify the types of cancer, prostate cancer in general, because that is a good segue to what genetic testing we do most often. So if we think about prostate cancer, at least 70 to 80% is considered sporadic, although that number may be decreasing as we learn a little more about genetic predisposition. Sporadic means that the cancer was caused by age or environment or lifestyle or chance, so something not inherited within your family. And that's the majority, and really age is probably one of the main contributing risk factors. So then we have familial, and that makes up another portion of the cause of prostate cancer. And familial, we can see in a family if there's prostate cancer clustering, so we might have one or two or three men with prostate cancer, and we haven't put our finger exactly on the genetic cause, but there's just too much prostate cancer in a family to think it's happening randomly. So we explain this by saying it's a family cluster. There must be some shared genetics that we don't fully understand, plus a little bit of environment and chance coming into play there. And then the last one is hereditary. So around 5% of prostate cancer can be attributed to a very specific known mutation that we have testing for, usually done through a blood test, to see if your family carries that very specific mutation, then we know those families are at the highest risk for prostate cancer. Remember back to that table, around 35 or 45% likelihood of prostate cancer. And that's helpful if we can pick that out in some families. That way the younger generations can be more proactive about screening, about prevention. I'm a visual person, so I wanted to show you what a family tree looks like. And I also would not be a good genetic counselor if I didn't show a pedigree. So pedigree is a family tree, and to give you orientation, circles are women and squares are men. And this line would represent a relationship, and then the line coming down would represent children. And then this line over here would represent siblings, and then we have parents again. So I'm showing you here a sporadic family tree. If I was in clinic and asked someone their family history and they said there's no one else with any cancer whatsoever in my family, and we usually go back to grandparents, aunts, uncles, first cousins at least, then we would consider this likely a sporadic prostate cancer. And also the age is important. So over age of 55 is helpful to know that it's more likely to be sporadic. This one is the middle group, that familial group. So you start to see a difference here. We have a cluster of prostate cancers in closely related individuals. So this would be the uncle to someone, these other family members. I meant to change these to prostate, but another way to look at this is you could have prostate, but if there are other cancers in the family, and do those cancers go along with each other? One of those is prostate and breast cancer. So this would be a family where we might be interested in, could there be this family connection? Nothing standing out to me from age, because all the ages are over age 55 or over age 50 for breast cancer. Skin cancer is fairly common, especially basal or squamous cell. So maybe this is not related to a specific gene, but it certainly is a collection of cancers, so we would call it familial. So here's the last one that I made very obvious. There's a lot of prostate cancer. There's a young man here with prostate cancer, and there's multiple generations. So this is telling me all the cardinal features we look for in a hereditary cancer family of early age of diagnosis, multiple generations, showing us this is something that's being passed on from one generation to the next, and just the sheer number of cancers. It would be highly unusual to have 4 prostate cancers in a single family and not have some genetic contribution. I propose this possible criteria for what is a hereditary prostate cancer family. And so by definition, we could use any one of these maybe to say if there's 3 or more first-degree relatives with prostate cancer, then we would be concerned that this could be something hereditary. If there's affected relatives in multiple generations, grandfather, father, grandson type of pattern, or there's young people with prostate cancer, and in that case, at least two relatives diagnosed less than 55. That would be a clue that the cancer risk is happening earlier. Could this be a genetic risk? So there's some well-known genes that we can test for when we're thinking about hereditary cancer. And I just wanted to put in here what is clinically available versus what is research, or what are we still understanding at this point. What is currently available from a hereditary prostate cancer genes, BRCA1 and 2, which I imagine many people have heard of before. It's most popular related to breast cancer and ovarian cancer risk in women, but it also is part of the spectrum of cancers for prostate cancer. There's a HOXB genes that are related, and then maybe CHEK2, which is also another well-known breast cancer gene, but we're starting to learn that breast and prostate cancer running in a family together could be linked by this gene. So we have clinical tests that we can order through doctor's office, through a genetic counselor, that analyzes these limited number of genes and can clearly say, yes, there's a mutation in a family, or no, that there's not. Then,
through a blood test?
And through a blood test.
It's a blood test.
It is also, it can be done through saliva or a mouthwash.
But it doesn't have to be the cancer itself.
It doesn't, it is definitely not the cancer itself. We are still, in that slide I showed you with the human body, and we looked at the inherited risk and then the tumor risk. These are still in that, what you inherit from your family. P53 would be in there as well. P53 is one of the major genes to help in the integrity of cells in general, and it can increase the risk for all cancers, but yes. The other, I circled the ones that are currently clinically actionable, we have good testing for, and we can get insurance to cover and pay for. All the ones to the right are genes or locations in the chromosomes that we think might have some role in the risk for prostate cancer. But testing for them is a little bit more on a research stand right now. So you see these names, and if you do any literature searches, you might see these pop up as related to an increased risk for a predisposition for prostate cancer, but we don't routinely test for those in families that we're worried about hereditary. So maybe I should define this. A SNP is called a single nucleotide polymorphism, and what that is just a single change in the DNA. We have found many SNPs that are related to cancer risk, but it's hard to quantify exactly how much risk those may have for a person, so that's where they're still in the research realm.
Under the HPC genes, if you have a test for one of those that shows you're predisposed, can you have a test for the other one that shows you're less predisposed, or will it kind of match up?
The way that these genes, their role is called autosomal dominant. So it means that usually a person under HPC would only have one mutation, We often draw one blood sample and test for the whole spectrum. So we might test for a list of 5 or 6. Then from that result, if we have a positive result in one, meaning we found the mutation, we have kind of found our main trigger for the cancer in the family. Even though all the other genes are normal, it doesn't help reduce the risk in this case. Good question. So I just wanted to spend a few slides talking about BRCA1 and 2, since they are the most common, and people have probably heard of those before. And then after a few more of these slides, then we'll switch gears and talk a little bit more about the tumor testing. So when they take your biopsy sample after you're diagnosed, and how do we use the genetic testing there. For BRCA1 and 2, it's the BRCA stands for BR is breast, CA is cancer. So it's breast cancer gene number 1 and number 2, and these were identified in the late 80s, early 90s because of a strong family history of breast cancer. But the more they studied it, the more they realized that it's really a spectrum of cancers: breast, ovarian, male breast cancer, and prostate cancer, and then sometimes pancreatic cancer in families. And the key here is if a family has a BRCA mutation, we don't focus only on one cancer type. We have the family start to screen for various cancer types, and especially the women are more proactive about breast and ovarian. The men more so on prostate, and then also doing some male breast cancer screening. I wanted to just comment a little on prostate cancer because there's a difference between BRCA1 and BRCA2 as far as risk goes. The gene has its normal role in your body is to help your cells correct DNA damage. And so both of those genes are doing that normally in your body, but if a person has a mutation in that gene, it increases the chance of cancer cells slipping through that normal protection. And the highest risk for prostate is seen mostly in BRCA2 families and less well-defined for BRCA1. But overall, if we look at all comers of prostate cancer, the odds of having a BRCA2 mutation in one individual with prostate cancer without a strong family history is pretty low all by itself. So I wanted to show you a very strong family history of a BRCA2 mutation. And so here's the prostate cancer, which is also, again, a young diagnosis. But then within the same family, breast, ovarian, pancreatic, and breast. So this is a BRCA2 family, and we know that the other men in the family, like this brother here, would be at risk for prostate and male breast cancer, even though the majority of the cancers in the family have been female-related. And then this male, remember squares are men, with breast cancer was a really big red flag. That's unusual to have a male breast cancer, and so certainly would have highlighted the need to do a genetic test. So I put here a little bit of guidelines of who should consider testing. One of the new guidelines that have come out in the National Comprehensive Cancer Network, which is called NCCN, it's what a lot of the oncologists follow, is that we should consider genetic testing in individuals sometimes based on their Gleason score. And a Gleason score of 7 or above is sometimes enough all by itself or with a small amount of family history to consider the genetic testing. And then, of course, if there's a suggestive family history. And people ask me all the time, I already have cancer. Why do I need to do this test to tell me what caused it or what I might be at risk for in the future? And the rationale that we try to explain is that we're using this genetic information for prevention. So to be proactive in the next generation for your sons or your daughters. So identifying the cause now can help maybe encourage someone who's younger to stop smoking or live a healthier lifestyle or start their prostate cancer screening at 40 rather than waiting till 50, which is typically when it would be offered. And then in the future, genetic testing, in not too distant future, could be used for personalized treatment. And so one of the examples right now is in ovarian cancer, in individuals that have a BRCA mutation, there's a new drug that is targeted only to carriers of that genetic mutation. And so we have lots of our ovarian cancer patients being tested for this to guide their treatment. And it's not too far off that we'll have very similar and many more treatments in the same respect. And this is all coming from the testing that's from the hereditary standpoint, so from the blood test.
And the
case of prostate cancer, would this be something that if your doctor says you should do active surveillance, you might want to do a genetic test first to find out if you're one of these people that is a high risk, because that might make the active surveillance much less likely to be successful? Just wondering about that.
I think when you say, asking about how do we manage or how do we do treatment based on genetic testing, it's really more geared towards testing of the tumor itself. So yes, your point is accurate. You should talk about that with your oncologist. I wouldn't ask for BRCA1 and 2 testing, but you would ask for testing maybe in your tumor versus testing of your genetics that you inherited. I wanted to include a slide here. This is fairly new. This is about direct-to-consumer testing. There's commercials. I heard one on the radio today for direct-to-consumer genetic testing. And I just wanted to say, a buyer beware type of thing here. Genetic testing is very popular and it's easily accessible now. There's one of the companies, I did list it, 23andMe is a popular company where you send them $200, they send you a saliva kit, you collect your own DNA, you spit into a tube, and then a few weeks they send you back a genetic result. And one of the risk factors they give you is a prostate cancer risk. So keep in mind, this is supposed to be for healthy people without cancer, and they're giving you a risk for what the chances are you might have prostate cancer, or the chances of actually a whole number of traits or risk disease. Heart disease, diabetes, male pattern baldness, all kind of funny things. But what I say about that is I would be cautious at this point about any test that's not involving your doctor or not interpreted by your doctor. So these direct-to-consumer companies, we call in the genetic counseling community, we call it recreational genetic testing. Because you could view it more as information and interesting and kind of fun, but not something that your doctor would act upon, at least at this point. I've done this myself, and it was really interesting that they told me I was supposed to have blonde hair and curly hair, and I thought, well, that's really not right. So, it's proof that it's not the full story. The Ancestry.com, all of those things, it's interesting, but you have to take it for what it is, and you get what you pay for. The clinical testing that we order is close to $4,000 to $6,000. So are you getting the same level of coverage and the same information for $200? So keep that in mind.
I do have a question about that, because 23andMe will send you the raw data, okay, for all the chromosomes that they check. What more would I get if I, got the kind of counseling that you would provide?
So the raw data is good. You're knowing what your genetic code says. But the problem is how to interpret that. So you might have one genetic variation, and if we did that same test in a clinical lab, they would be able to tell us, is that mutation well known to be harmful to the protein, or not harmful, benign to the protein, or uncertain? And it's hard to tell that just from the raw data, sending it through a third party to interpret. The other thing is, let's use BRCA1 and 2 as an example in 23andMe. So 23andMe will test for BRCA1 and BRCA2. Difference is, in clinical testing that you would go through your doctor or a genetic counselor, they're checking for close to 4,000 different genetic variants in those two genes. 23andMe tests for only 3, the 3 common that are in Jewish ancestry. So it's not full sequencing of the genes, and you would be missing out on incomplete information.
A lot of these diseases are not all caused by a single gene, it's a combination of genes. So
Polygenic. Right.
So you put this together, is it pretty well known what combination is necessary to cause the disease?
So that's where we're lacking. We know what single genes can contribute to a small percentage. But do we fully understand what these polygenic risks are? So if there's a test that looks at 10 risk factor genes and you have variations in all 10, and how do they combine those? And to my knowledge, there's no good test that's been validated that can actually give you an end result type of number.
So you don't get all the poly you need to have blonde hair, right?
Right.
You got part of it.
It's hidden in there somewhere, my genetic test told me, but something else was impacting that. So I think the point is it's very complicated, and technology is great because now we can sequence your whole genome, and it used to take months to sequence one person's full set of DNA, and now it can be done within 24 hours or less. But just because we have the data doesn't mean we know how to interpret it or how it works in real life. Okay, so a little more about, all of this in the past here we've talked about is hereditary. That small percentage where there's a specific mutation, we're tracking it through a family, we're thinking we're figuring things out. But how do genetic mutations impact how cancers grow? So why does a normal cell become a cancerous cell in the first place? And that's what I wanted to spend most of the rest of our time talking about. So if you have one definition for a cancer, it's uncontrolled cell growth. So you have a normal cell, and suddenly it's gained this ability to grow when it's not supposed to. And all cancer is genetic. So all cancer has genetic mutations within that cancer cell. But not all cancer is inherited. So what I mean by that is, if we take a biopsy of a prostate cancer, we're going to find genetic mutations. That's what made it become cancerous. But those mutations were acquired over a person's lifetime, and usually, and at least in most cases, we haven't figured out all of the genes, the polygenic effect of which ones were inherited. So I thought I would put up here, what does it take to become a cancer cell? And there are three main things. A cancer cell must keep growing. That's like its mantra. That's what it wakes up and does every morning. It must keep growing. So there's something that's called Proto-Oncogenes. Which I have a typo there. Whoops. Proto-Oncogene is a normal gene that's kind of like the gas pedal for the cell. So think about your cell as a little car going down the road, and if it needs to divide itself, it steps on the gas and it can create a new version of itself. And that's normal. Your cells only have a certain life cycle, and they need to replicate sometimes. If there's a mutation in one of these gas pedal genes, it's called an oncogene, and then it's permanently pressed down. So you're permanently going 90 down 75. And that's where the cell keeps growing when it's not supposed to. A cell must lose the ability to fix its problems, or it must lose its ability to stop its cell cycle. So you have a gas pedal, but you also have a brake system, and your cells should know when they have DNA damage, and they would put on the brakes, and so you need those genes to work normally. But if you have a mutation in your brakes, now all of a sudden you're pushing the brakes and they're not working, so the cell is still going forward when it shouldn't be, still dividing. And then finally, cells gain the ability to move where they're not supposed to, metastatic disease, and to grow their own food supply. So wherever, if prostate cancer grows so big, or if it spreads somewhere else, it has to have the ability to get nutrients. It gets its own blood supply and oxygen and those things. So those are the cardinal features of what makes a normal cell different from a cancer cell. And all of these, by the way, are controlled in most part by genetic risk or genetic mutations from what's normal. So I like this picture. It shows you a normal subset of cells. Here's some little cells in a petri dish doing their normal thing. And if you think about it, over time, just because we're humans and our cells make mistakes, you could think about any of these color changes as a genetic mutation in a cell. Sometimes it's the gas pedal, sometimes it's the brakes, sometimes it's the growth factors. But as long as the other integrity of that cell is maintained, we can keep those cells under control. So really, at this point, we're all making cancer cells, but your body has these normal mechanisms to help prevent them from growing any further. But as time goes on, we get older, we're exposed to smoking or alcohol or certain dietary risk factors. The mutations can become more so until finally you might have one little cell that has a mutation in all of those normal breakpoints, and that's where the cell that would take off and really grow into a cancer. Just for a comparison, hereditary cancer, you can kind of think about it like someone inherits that first mutation in all of their cells, so they're starting one step ahead. So it takes less time for them to develop a cancer in these cases of hereditary cancer. So I think some of the comments before I came, the emails I got or questions I got, asked about there are mutations in all prostate cancers, and can we talk about some of the common mutations and their driving role in cancer. And I thought I'm probably not the best person to explain those, but I can at least give us a framework to work off of, and then an oncologist would be able to give more detail. What I wanted to show, and it's really tiny so you won't be able to see it, but really the point is we know all tumors, all cancers have some genetic mutations that are acquired, so they happen over time. And you can't see it, but prostate cancer lives right here. So what this is showing us is on average, there's around 40, 50 in this range of mutations in any given prostate cancer. And what science has learned over time is that a lot of these mutations happen early in life, but they just kind of hang out there and they're not doing anything bad until there's one major mutation in a gene, like the P53 gene, that's important for cell growth. And that one mutation is what maybe drives the majority of that cancer turning from a precancer cell in your 20s to a full-blown prostate cancer in your 60s. I wanted to point out this difference, this language you might read about driver mutations or passenger mutations. So this is in your tumor. What they know is there's maybe around 50 mutations, genetic mutations in a tumor cell, and the majority of those are just passengers. So they're a byproduct of being cancer itself. Cancer causes more and more DNA damage. So a lot of the gene mutations don't cause direct advantage for that cell to grow. But a driver mutation is fairly small number, so 2 to 8 per cancer. And those are ones that we know are actively causing the cell growth. And they're in pathways that we can target with treatment. So think about this. If you know which gene is causing your brakes to fail on your car, if you know that, I'm not a mechanical person, so bear with me on this. So if you know what is causing the problem on your brakes, and you go in and fix that, now you might have a way to combat that cancer cell. And that's a way you can think about personalized therapy or targeted therapy, is we want to do a genetic test on a prostate tumor, figure out what mechanism is broken and is driving that cancer, and then hopefully there's a treatment that can target that exact pathway that's missing or messed up. The whole goal here is, at least in testing of the tumor, is using genomic information from the person's actual cancer, so very unique information, to personalize medicine and decide which treatment to use. So just like every person's different, every cancer is different. Yes, we call it a prostate, but I know if all of you shared your own story, you would tell me what stage and what score and how far advanced and those type of things. And those are differences in the types of prostate cancer that you have, but also there's a genetic profile that's unique in your cancer. So if we can understand more about this biology, then we're able to hopefully reduce side effects by picking drugs that work better but cause less of the symptoms, and also pick out patients who are going to receive the best treatment. So this all sounds really exciting. I think I've convinced you that this is a good way to go and this is good research. But the problem is we've got a lot of technology, but it's really hard to interpret how it's best utilized in a group or for any individual person. And we can do research and look at a group of 1,000 men and say the genetic mutation that was most common is this mutation, but then we might not have a targeted therapy that works for that mutation. So we're really in a period of a lot of research and growth. And I think the NCCN, which I mentioned earlier, has guidelines for prostate cancer. So I pulled this from their guidelines earlier today. So I think it shows where we're at in the use of this technology. They recognize that there are several tissue-based molecular tests or genetic tests that are used for newly diagnosed men to consider active surveillance or to consider treatment. And they also can use them in the metastatic setting as well. But right now, what we're basing this information on is a lot of retrospective data. So it's where they take men who've already been diagnosed and had treatment, they go back and look at their genetics and try to figure out if their treatment was somehow guided by the genetic testing, so it's retrospective. There's no randomized controlled trials to say, okay, we have a group of men who are newly diagnosed, we're going to put part of these men to do genetic testing and do their treatment based on that, and part of these men will have the standard treatment, and we're going to watch their outcomes and see which one is better to prove that the genetic information is useful. But they do mention this will be increasing in the years to come, so there'll be more tests, they'll be better validated, and we've found mutations, but now we have to figure out the drugs that can treat those mutations, and that's really where we're at now. I think some of the tests that, hopefully I listed them, yes. Okay, so I listed them here. These are the ones that I've heard of in our oncology clinic, but I have to honestly say, as a genetic counselor, I focus more on the hereditary part. I'm on a work group for TriHealth where we evaluate these new tests, but the oncologists are really the ones that order it because they're the ones that are helping you and your families decide about active surveillance versus more aggressive treatment versus what other options there are. So that's where I would say my understanding kind of stops here, as I can get us to this point in building our case, but to say whether any individual person needs to have a Prolaris or an Oncotype test, I don't think I'm the right person to say that.
Program is to treat, so it's not a test, right?
Yes, so sorry, I put that under there as an example of a personalized prevention or a personalized treatment or therapy. Okay, so conclusions, and this is kind of putting everything together from the hereditary and the tumor testing. So I hope I proved it's important to know family history, since that can be a clue to risk if someone has not developed prostate cancer, maybe a clue of who is at highest risk. There's definitely a difference between the genes that you were born with and the genetic mutations and the genes of a cancer. So we always expect to find more mutations in cancer. Use caution when you're reading about advances in genetic testing. It's often the technology that's ahead of what we can clinically use, so the word clinical utility and things like that. If you go in and try to look at PubMed or some of the medical literature and you're reading conflicting information where there's a certain biomarker or a certain genetic test that showed to effectively predict who had more aggressive prostate cancer in one study, but then in the next study they couldn't replicate it because it was a different type of population or they had some difference in their study, that's a clue that we're not quite there yet to figure out who these tests are really working for. And then the last one is related to the hereditary testing. So who should consider genetic testing? I put if you have a Gleason score at or above 7 and maybe one of the following, then you would consider meeting with a genetic counselor or asking your oncologist about the hereditary genetic testing, like the BRCA1 and 2, the CHEK2, the P53. And also if you have prostate cancer and there's a breast cancer, or ovarian or pancreatic in your family, even if it's a cousin, even if it's a grandparent, that may be significant enough to try to offer the testing. Sometimes we find BRCA positive families where there's a small family. Nowadays, we only have 1 or 2 siblings in each generation, so that can be masking a family history, as opposed to families where I grew up in southern Ohio that were big farming families and we had 10 or 12 siblings. Those are genetic counselor dream. But they don't show up anymore. Okay, so where do you go for questions about testing? And I want to differentiate myself as a counselor, master's degree versus oncologist, even versus maybe geneticist or a PhD genetic scientist. So go to your genetic counselor if you have questions about cancer in the family and should you have testing or not, blood test. Go to the medical oncologist if you're thinking, do I need that biopsy of my prostate analyzed to see if I need a targeted treatment or how aggressive we should be. And then we're going to leave all of the genetic studies and figuring out the pathways and finding us new drugs to PhD geneticists and scientists, and hopefully those will be forthcoming. So that's the slides I have, but I can stay as long as needed for questions, and I also printed questions that I can read if that is helpful too.
I posed a question online, but I'll ask it here as well. So my father, who died of prostate cancer, I guess he had an aggressive form of prostate cancer. Does that mean that my prostate cancer is also an aggressive form?
Not necessarily. And so your individual prostate cancer is going to be, the aggressiveness is going to be based on the molecular biology of your tumor versus your father's tumor.
I had an Untitled TX test, so I've got a score. I'm trying to make sense of what that means.
And I wish I could help, but I've never interpreted one. And has your oncologist been very helpful?
Well, in a brief statement, basically he says this is the percent chance that it's a level of aggressiveness. So it's a percent chance that it will not metastasize. Is basically how they explain it.
Mm-hmm.
And so, of course, the opposite is the chance that it will. And I'm assuming they did that, like you say, retrospectively, looked at thousands of people who got a similar result, and some percent metastasized, some didn't. I'm assuming that's how that was done. The only thing about statistics is it doesn't make a difference if it's you, it's 100%. So I've got this statistic and I don't know what to do with it.
I can relate this to, we use an Oncotype, same lab, same company for breast cancer patients, so we have more data on that. And I can also relate it to when I talk to families about the hereditary testing. It's almost like you have to have an internal threshold of what your limit is. If they tell you 10% or more, then you're going to act on it. If they tell you 20%, 30%, but I don't think there's enough information to guide and tell you exactly what percentage feels right to you to make the next step.
Okay, I have a couple of questions, but one of them is about, let's say you have a genetic test and it finds that you have an active mutation, like you say, a driver type gene. How do they, is there some way to understand the level of expression of that gene? In other words, are all of these driver genes equally aggressive, or do some express more than others, and if so, how you determine that?
So not clinically available to interpret, that type of information, but yet on a molecular level, yes, if we have a patient who has a Foundation 1 testing and we get results back, and we find that there's a driver mutation that is known to, severely impact the function of cell division, then we know that we may treat that one perhaps more aggressively. But it would still be standard treatment, standard chemo or radiation or surgery of what they would normally do. There's no targeted therapy to it. The other thing we have not covered is tumor heterogeneity, which means if you have a tumor, you could take a biopsy and get some mutations on one side, and then you might get other mutations on the other side. And so it's very difficult to make a decision if you feel like you don't have a complete story on the whole cancer. But sometimes we'll get a percentage from these type of tumor tests that will tell us the allele frequency, so the percentage of cells and DNA in our sample was over 50% for a mutation, and that means that it's probably a large percent of the tumor has those mutations. And if it's an active mutation we can target, then that's a good thing.
Yeah, I guess the reason I'm asking is because some of these, again, you're perhaps not cluing in on mutations in the tumor, but some of these genetic mutations may be present, but they don't always seem to be correlated to Gleason score. I could say, like, a large population of tumor cells contain this one mutation. But it occurs both in Gleason 6s and Gleason 7s and in Gleason 8s, maybe a little bit more in Gleason 8s. So this raises, to me, always raises a question about whether all Gleason 8 tumors are the same.
Right.
And there doesn't seem to be a good answer yet. But I guess the reason I'm asking is about, there's genetics and then there's what they call epigenetics, right? Our ability to control our destiny.
I knew you were going to ask that.
Okay.
Go ahead.
So, I mean, rather than just being the victims of our genetic profile, what can we do about it?
Okay, so what is epigenetics? Epigenetics is, we cover genetics. That's where there's DNA damage in the actual strand of your DNA. Epigenetics is the environment, which could be drugs or, pharmaceutical drugs, diet or lifestyle or other affecting agents, hormones that surround your DNA. So they are in the nucleus inside of your cell, and somehow chemically they impact which genes are expressed and which genes are not expressed. So I brought this slide up to show you, this is similar. Here's a chromosome, here's this DNA strand. Epigenetics helps on this level tell your DNA to unravel and encode itself, or keep really tightly bunched and not express itself. So it's a really complicated thing. And your thought process is right on, in that we should be able to modify our diet and our lifestyle hypothetically to help control what we can. We can't change the structure of our DNA at this point, but can we change the environment that are surrounding these cells to help reduce genetic mutations or help offset maybe a mutation that causes a cell to divide too much? Can we do anything to control that from the environment of your DNA? To my knowledge, there's nothing from a diet or a lifestyle standpoint that could tell us quantitatively, if you do this, you'll have a reduction by X amount. In general, just like all the doctors would tell you, exercising, eat fruits and veggies, lower fat is all good, but there's no quantitative evidence that tells us how that would work or what the protection is. Lots of research on it, but I don't feel like I can comment intelligently on it.
I have a question.
Sure.
And I don't know if you can answer this or not, but is it better to talk to your doctor about this when you're newly diagnosed with prostate cancer? Because, like in my husband's case, he had surgery last August, and they came back and said that it had already spread outside the prostate and everything at that time. So they took his Gleason score up from an 8 to a 9 at that time. And nobody talked to us about any type of genetic testing or anything back then. What if we wanted to do that now? I mean, do they keep samples? How long do they keep samples that they could go back and test like the original tumor? Or would he have to go back in for, some other types of tissue testing?
Any extra procedures?
Yes.
So first part is, when should you ask about this? And I don't think that it's ever too late. So if you're newly diagnosed, yes, of course, bring it up and see what your doctor feels is the current state of the tumor testing or the hereditary testing. But if you're later into a diagnosis, it's still a good time to talk about. And then the other question is what type of sample do they need? So from the tumor samples, they can often run this from a pretty small biopsy. Most hospitals will save biopsy samples. They're legally required to save it for 10 years. And so I would say when we get a few years later, sometimes the quality of the DNA is not as good, but it's still there. We could try it. And then the labs that do these tumor tests would try to run a sample, and if there's not enough quantity of cancer cells, or if the DNA is not good anymore, then that's when you would have the option to maybe do another biopsy and get more tissue.
Thank you.
Sure.
This might be a good point, it's not a question. A couple of weeks ago, I heard a similar talk about breast cancer. And something that got mentioned there that I had not heard of. There is a tumor bank. And I'm mentioning it because we all with cancer should be contributing to this. They take the parts of the tumor that the hospital doesn't need to keep, because they only need to keep so much, then could go in the tumor bank. And the reason this is important is that all of these gene researchers, instead of having to wait years to get enough prostate samples, could go to a tumor bank and instantly have 100 or whatever. It's nothing that any of my oncologists ever told me about, so I'm trying to get the word out. And there's no cost. It's not like we don't pay to do this. This is for researchers, so that down the road, both for us and our children, the research can get done faster.
I guess on a related note, there is also DNA banking from a blood sample that's able to be done too. So if we're worried about a hereditary risk and a family has all normal genetic testing, sometimes a person will bank their blood sample to do genetic testing later on, and that's fairly reasonably priced. So it's not research, as you mentioned with the tumor, but to bank a person's own DNA is about $100, and they'll save it for 20 years. That way in 10 years, if there's some new test and your grandmother is no longer living, then we can pull that back out and maybe run the genetic testing on the person who had a cancer to see what the hereditary risk factors were. So that was a good reminder for me. Sure.
I was at Children's Hospital about 5 years ago. They had a genetic test done. I was already having problems, and since then I've had the Da Vinci prostatectomy. That was last May. But anyhow, it's obvious in our family, you know, the men and the women both. The sister just died 6 weeks ago of ovarian cancer. 2 sisters had breast cancer. And my uncle's grandfather, prostate cancer, you know. So it's really a problem. So anyhow, I get extreme pressure from the family, thank goodness, about having all the tests done. And my sisters especially are real definite about me getting a mammogram. Now my family doctor and even Dr. Kuhn that did the prostate cancer surgery just thinks this is absolutely ridiculous, and it will come up like in another month or so. My sisters will be there. Mike, you haven't done this. Well, I have tried to find where you can have something like that done, and I can't. So like in your department at where you work, do they do this? I mean, where do I go for this to get my sisters off my back?
You are a great brother. So yes, it's, you know, we don't talk about it often, but male mammograms we can do, and they do them at Bethesda North in the breast center there. You know, it doesn't take any special machinery. Really, I think any hospital around the city would be able to do it. It's a matter of getting your doctor to put in the order, but then it's just done like the same procedure as women would do.
I'm not sure my doctor would put in the order because they just think this is ridiculous. And maybe it is because of the occurrence of it. And you know, in my family history, since this is so strong, I doubt that my uncles and grandfather are going to talk about this as they're sitting around on Sunday afternoon drinking a beer with all the rest of the guys. But, you know, my sisters are real definite about me having this done one way or the other, finding out where I should do this.
We make that recommendation too. If there's a known mutation and there's a strong family history, we have, you know, families be as aggressive as we can with the screening. And a mammogram is not invasive, so there's, you know, not a needle involved or anything. And so, you know, if it's something reasonable to you, that's part of the spectrum of care for a gene-positive family.
Okay, so insurance covers this, probably? It's like good insurance, Medicare, or otherwise. Let me ask you this too, as far as like in my family, because like the women were dying of breast cancer in their 20s and 30s. That's how bad it is in my family. And so men can have pancreatic cancer. And so with me, is that something that maybe, I mean, with longevity is in the family for people that don't get knocked down by these problems. But you know, like, I mean, is this something eventually I will get because I have prostate cancer or not?
It would be unrelated to your prior prostate cancer, but would be related to that genetic predisposition. What we typically know is that pancreatic cancer is more common in BRCA2 families, so that risk is up to about 8% lifetime. Pancreatic cancer screening is not very good. We don't have a good detection method, so really you have to work with your doctor and say, I'm at this level of risk. And if you have a close relative that had pancreatic cancer, again, we would probably offer some type of pancreatic cancer screening. Versus a family who's fairly large, has had other cancers but not pancreatic, then it's not as strong as an indicator. But that's where, you know, you said you went to Children's.
I did, yes.
And even though it's been years ago, if you need support in saying, hey, my doctor's not doing what is recommended, by your sisters, but really, could they put it in writing for you to help your doctor justify ordering it? And the same type of idea with pancreatic cancer screening. The first step is just talking with a GI doctor, because they're the ones that do these screenings all the time and make that diagnosis. So they're the best ones to help you figure out what are the risks of a pancreatic cancer screening, what do I have to gain by early detection, and then kind of make that choice.
Okay, well, I probably will get a hold of your department because I've already dealt with the doctors.
Fine, work for you.
Okay. Because I think it's kind of unfortunate, like, that they don't want, do something like this, just approve the test to be done, but they won't do it.
We help, genetic counselors help in that in-between. So now we have a diagnosis, we keep up to date on all the recommendations for management, and that's really hard for the primary care who's managing a whole bunch of other things. So maybe it's getting in touch with that genetics department, having them kind of relook at the case and forward the current management guidelines to your doctor so we can say, here's the proof of why we're recommending this, and that gives support for things to take off.
I'll be in contact with your office, like when you're finished, if you have a phone number or somebody I can call about this specific problem of mine, because I have another 6-month, exam by Dr. Kuhn coming up in a month, and so, I'll talk to him about it again.
Sure.
Yeah, so. But anyhow, thank you very much.
When my daughter was born, I considered being cord blood. I didn't do it, but that's like $1,000, and I wasn't sure what good it was. If you can get all the information you need from spitting in a bucket or from taking a little blood from your arms, what's the good cord blood? What is the idea there?
Cord blood is a little different because they're cells that have the potential to become any other cell, whereas your blood cells are already differentiated, and so we can get DNA from either, but really what it is, the cord blood allows you to do transplants later on in life. So for a blood disorder or something where someone needs to have a bone marrow transplant, you can use your own cord blood, or there's other rare genetic conditions where the cells themselves are what's useful, it's not the
Only way you
DNA.
can get undifferentiated cells in order to...
Well, there are probably other ways, but I'm not the expert on that.
I should have done it or not.
I didn't do it. It's kind of like insurance or the lottery or odds. If you have, as far as banking goes, if you have a rare genetic condition, not cancer, but anything else, or if you have a leukemia or lymphoma in the family, probably makes sense. If there wasn't anything like that in the family, then the odds that you would use it might be lower.
I have 2 different questions. The first one, what the gentleman was asking about his father. So between your father and the son, or between 2 brothers, you say there's just not enough relevant matter to draw some conclusions of one cancer from what the other brother experienced?
As far as type goes, aggressiveness and those type of things.
Right.
Correct.
You can't draw a conclusion. Okay. As far as aggressiveness and changing something from a low aggressive to a higher aggressive, I've just become aware of these warnings about vitamin B6 and what is it, vitamin B12, maybe some of the others. Are these, are they, like, people have the 5-hour interviews at work and stuff like that. I think those are kind of loaded with vitamin B6, aren't they?
Mm-hmm.
So for someone that has early stage non-aggressive prostate cancer, is that something they should be staying away from completely?
Good question for your oncologist.
Okay.
Well,
Well, I mean, like, I just became aware of it recently,
Sure.
It was actually in the pamphlet that they gave out. That would even be like a vitamins have 1 a day, vitamins have B6 in them. I mean, is there any, you're talking about the drivers, so you don't see any evidence that they are a driver?
No, I mean, genetic drivers, unrelated to a supplement, maybe an epigenetic effect. So, you know, I would say if your doctor works with a nutritionist, that's what my go-to person as part of our services, have nutritionists that would probably be the most up-to-date on that. Everything in moderation, so, you know, these energy drinks are loaded with it, but if you feel like you need to cut something out, I would check with the doctors.
Yeah, and you know, the kids today, these 15, 16-year-olds, they're drinking the energy drinks and stuff, and it's got all this stuff in it.
Yeah. All kind of chemicals.
Yeah.
Can you talk anything about, I don't know if this is relevant to this topic, about the Decipher test? Are you familiar with that?
Decipher?
It's like,
It's DECIPHER. My husband had it. Tell them what they did.
Don't do the genetic test like you're talking about.
So probably in the same range as that Ontitled Foundation one, Polaris, just a different name, different company, but maybe same idea.
And trying to determine whether how much of a risk you are, whether you should get further treatment down the line or not, based on what they found.
And for him, they said that if he needed treatment, he should definitely get it because it indicated that he might get bone cancer. The Decipher test indicated that. If something had reoccurred, that he would need to do radiation or something more aggressive.
So that's most of these tests that are on the tumor are analyzing multiple genes or looking at the expression of multiple proteins and then combining that score, and the end result is often a number or a percentage or something, but it's not a yes or no. So it's not going to tell you, yes, you definitely should, or no, you definitely shouldn't. It gives you this range, and then it's left for you and your doctor to kind of talk through and say, I'm at a low, intermediate, or high, or whatever, however they classify it. And that can be a struggle because then you're wondering if you're making the right choice. You know, we have this fancy, expensive test that's supposed to be the answer, and then it still leaves questions, and I think that can be difficult too.
well, I think, if I may, that's about exactly what I asked you, because according to Google, the Decipher test measures the expression of these biomarkers, of these 22 RNA biomarkers, to calculate the probability of clinical metastasis. So,
Can you talk a little louder?
sorry, I don't want to take over, but this Decipher test measures the expression of these biomarkers, or these genetic biomarkers, to assess or calculate the probability of metastasis. So that's what I was asking you about, the levels of expression.
So it's a combined score, it's not one at a time.
Yeah,
Right.
but I mean, the level of expression, I mean, isn't that, I think that's where the,
I mean, so the platforms of testing that they do can look at individual mutations, they can look at protein expression, and how overexpressed or underexpressed. So they know what the normal level should be. If you have one gene coding one protein, that would be the normal expression. They can tell if it's overexpressed. But the key is that overexpression meaningful? And is it meaningful packaged all in one test? How accurate and how valid is that? And I think that the technology is there to do it and give you a expression or a level. You can say one protein is 2 times normal or 5 times normal. But how valid is it?
is it 1 or 2 times, or is it 100 times? I mean, like what are the orders of magnitude involved here?
So when I asked, and I'm preparing for this, when I asked our oncologist at TriHealth, I asked 2, I said, do you order this routinely? And they said no, they kind of reserve it for just few cases, metastatic disease maybe, or something like that at this point. But what I'm hearing you guys say is maybe it's offered early on to decide about active surveillance versus more aggressive treatment.
Well, I think not. I mean, I think, at least according to what they tell us, it's for men who've had a prostatectomy, and it's a predictor of the likelihood of metastasis post-prostate?
I mean, I would say the oncologist would be able to answer that. I can go based on when I know in breast cancer and more validated tests, there's a protein expression called HER2, and if that is 2 or 3 times normal, that's not as aggressive as maybe 13 times normal. So biologically or plausibly that would make sense, but can I say that's true for every biomarker for prostate cancer? I don't know the answer, and I don't know if there is one.
Yeah.
Other questions? Well, thank you so much for having me. I've enjoyed the group.